Hola. Somos.
Somos una universidad católica regional. Más del 80% de nuestros estudiantes provienen del Maule y más del 80% de nuestros egresados se quedan en la región. Somos parte del desarrollo del capital humano avanzado, fomentamos el capital cultural y generamos proyectos que van en directo beneficio de la sociedad. Desarrollamos el espíritu de servicio y el pensamiento crítico a través de nuestros planes formativos y sello valórico en pregrado, posgrado y educación continua. Somos parte de la historia de 15.000 personas que han pasado por nuestras aulas y hace más de 50 años que contribuimos con el desarrollo de la región, siempre responsable y comprometida con nuestro entorno. Contamos con programas de acceso inclusivo, porque creemos que no hay impedimentos ni sociales, económicos o físicos para concretar los sueños. La investigación que se genera dentro de la UCM nace desde las necesidades locales. La innovación busca cambios positivos en la sociedad, dando respuesta a los requerimientos de nuestra zona. La UCM es fiel a sus orígenes, pero mantiene su vista en el futuro. Somos tu universidad. Somos una universidad católica regional. Más del 80% de nuestros estudiantes provienen del Maule y más del 80% de nuestros egresados se quedan en la región. Somos parte del desarrollo del capital humano avanzado, fomentamos el capital cultural y generamos proyectos que van en directo beneficio de la sociedad. Desarrollamos el espíritu de servicio y el pensamiento crítico a través de nuestros planes formativos y sello valórico en pregrado, posgrado y educación continua. Somos parte de la historia de 15.000 personas que han pasado por nuestras aulas y hace más de 50 años que contribuimos con el desarrollo de la región, siempre responsable y comprometida con nuestro entorno.
I'm sorry, I will just not do that.
Okay.
Bueno. Ricardo!
We are going to introduce you first, and then I will
Cheers.
give you with your lecture. Okay, vamos a comenzar el simposio de hoy entonces, que se titula Metabolismo en espermatozoides y en espermatogénesis. Y tenemos dos invitados para este simposio, que es el doctor Joao Ramalho-Santos de Portugal y el doctor Ricardo Moreno de Chile. En primer lugar, entonces, voy a presentar al doctor Ramalho-Santos. Yo no sé en qué idioma va a hablar el doctor Ramalho-Santos, si en español, si en portuñol, si en portugués o si en inglés. Lo voy a presentar en inglés para que él lo pueda entender bien. Dr. Joao Ramalho-Santos is professor at the Department of Life Science, University of Coimbra, Portugal. He got a PhD in cell biology in 1997, and also he has a postdoctoral fellow in reproductive biology at Oregon Health Science University in the United States. And he is a visitor science at the University of Pittsburgh, USA. And he is a Fulbright Fellow at the University of California in San Francisco. He currently is the head of the Biology of Reproduction and Stem Cell Group at the Center of Neuroscience and Cell Biology at the University of Coimbra, and he works in bioenergetics and metabolism of gamete and stem cells, and on science, communication, and bioethics issues related both to the assisted reproduction and metabolism, aging, and disease. He's an associate editor of several journals, and he's going to talk us about human sperm metabolism from the clinic to biochemistry to outreach. Dr. Ramalho-Santos, thank you very much, and you may begin your presentation. Thank you very much.
Thank you very much. Thank you very much for the invitation. I'm sorry I'm not there, but hey, we'll do the best we can. Can you hear me okay? Should I speak louder and or slower? Is it okay? Okay, good. If nothing else is said, I will just move on. So thanks for the invitation. It's a great pleasure to be in this meeting. As the kind presentation of my career, here it is in one slide. So this is where I've been training and throughout my career. I would say that my postdoc at Oregon Health Science University is where I met actually the second speaker, Professor Ricardo Moreno, and we have many, many papers in common from that period. So I work in more of a biomedical complex. So we all work in kind of bubbles, what I call disciplinary bubbles, because it's so often difficult to talk to other people from different fields. So my focus is on the biomedical sciences. They're in green in the center of the slide, a bit on clinical medicine. We have also some environmental concerns that the projects that we work on. But since we work with human patients most of the time, we also work with psychology, communication, ethics, and other sorts of issues. That comes from
Dr. Ramalho-Santos, can you interrupt you for a moment? Because we are not seeing your presentation. We are seeing your camera. Can you make small your camera, please?
Small my camera, yes. Oh, I see what you mean. Like that?
No, we're still seeing your camera and yourself.
Why? I'm not seeing it. Okay, stop presenting. So share again. Oh. What about now?
Okay, now it's okay. Now it's perfect.
Sorry about
Thank you very much. I'm sorry to interrupt you.
Oh, feel free. I mean, I could be talking to a wall for all I know. So yeah, so this is the kind of issues that we deal with. In my lab, we work with many things. So we work with sperm, oocytes, ovarian tissue, testicular tissue, and also stem cells. And in your careers, for younger members of the audience, if you work on many things, what your reviewers will tell you is that you're not really good at any of them. So you have to focus somehow, which is what we did. So we focus on the same things related to all these systems. They have to do with metabolism, with mitochondrial activity, with reactive oxygen species, and other such aspects in all these systems. So I will focus a little bit mostly on sperm today, but we also work on other systems. And of course, in sperm, it's related mostly to male infertility and assisted reproduction. So we work also in a hospital context. And we work for basic biochemistry, from a basic biochemistry standpoint, with mitochondrial memory potential, reactive oxygen species production, ATP production, and other metabolic pathways that are involved, one directly or indirectly, with this. And we use for this different sorts of techniques, such as the fluorescent probes that monitor different things. We use a bunch of them for all sorts of mitochondrial properties, both for memory potential and reactive oxygen species or nitrogen species production. And we use both flow cytometry and cell sorting, which we have the facilities at the hospital to do. And we also use fluorescent microscopy and for more simple things, and we have different probes for different aspects. The important thing that we've learned in this study, and that's also a message that is quite common, but people often forget it, is that when you start working with an antibody or a probe or a technique, you read the fact sheets or the instructions, so to speak, and well, then you apply it to your own cells and it doesn't work. So what we found in our work, and that's actually, it gave one PhD thesis just on this topic, was that that in sperm and human sperm, and using our protocols, some probes that we thought that were marketed as being sensitive to hydrogen peroxide or superoxide were actually not sensitive to those things, or not only sensitive to those things, and therefore, at least in sperm, we're not monitoring what the company says they were supposed to be monitoring. So it's always important to do controls and be aware of what you're doing, of what you're getting. More recently, and this is a paper that we are preparing for publication now, we have used the Seahorse flux analyzer. Maybe for those of you who don't know, it's basically a system, it's basically a pH meter and or coupled to an oxygen electrode. And what it does for the OCR measurements, which is the top portion of the slide, It monitors what your cells, sperm, oocytes, stem cells, what have you, what they're doing in terms of oxygen consumption and ATP production using mitochondrial-related processes, or using the ECAR protocol, which is at the bottom of the slide, exactly what they're doing, your cells, sperm, whatnot, what they're doing in terms of glycolysis via acidification of the media and lactate production. The top part is via oxygen consumption. So these are also things that these are also techniques that we have set up in the lab, and we're doing it with different populations of sperm from different types of patients, normal zoospermic patients, and other sorts of patients with low counts, sperm counts, low motility, and other sorts of aspects. I'll get into that. So human sperm. It's a cell, which is very weird because its function is outside the body it's made in, and not many cells are like that. It's transcription and translation inactive, or that is what is thought, at least for the most part. It can get proteins and other cell sort of molecules from its environment. It can capture things, but it's mostly what you see is what you get. There's a small cytoplasm, which makes it very hard to work with the cell in several sorts of techniques. I have a student now doing patch clamp on sperm, so electrophysiology, and due to the low amount of cytoplasm, this is an extraordinarily difficult cell to patch. There are just basically 3 groups in the world that do this, and she's working with one of them because I thought that was the best. And we have the basic clinical dichotomy, which in this field, which in this part of my group, we deal with every day, which is that we want to understand things, basic things about sperm and how it does, it performs its functions and what proteins are important, what metabolic pathways are important. The clinician, however, is not interested in any of this. Because that's the leader of the group, the medical group, they're interested really on practical. Can we get better success rates? Can you improve this and that and the other? And that's what we have to do. So we have to kind of balance this here in our interests and interests of the clinician. And this has to do with applying what we know to improving the success of assisted reproduction techniques. So we have been able to do this in our group and in the hospital, which led to the hospital being kind enough to hire people, hire biologists and biochemists because they thought, well, and basic scientists, because they thought this could actually help them, and we have numbers for that, not just we think it can help you, we can show it can help you. So that's important when you're trying to move towards a more practical application of things, which was not at the beginning was not my interest at all, by the way. By the way, since people often ask me, so what do you do on the female side? On the female side, we have a very important, we are the leading group in Portugal for oncofertility. So that is preservation of ovarian tissue from women who undergo chemotherapy and then may become infertile, and then we transplant the tissue back. So this is something that we do in the lab as well. I won't talk about this at all. And also oocyte aging, which is has to do with maternal aging. And due to the clinician asking us to be involved in this, we also have developed models and studies to improve the quality of oocytes from aged women. But I won't talk about this either, just to let you know that when you have to work in a clinical environment, you often have to have your own interests, but then you have to be available to help in other ways. Okay. So what do? We address cases of male infertility, and we try to understand why the sperm works or does not work. Interestingly enough, some of our findings have been interested to pharmaceutical companies who have contacted us not to improve the success of assisted reproduction, but to participate in the evaluation or development of male-based contraception strategies, which we're also doing. And I won't talk about that too much either because, well, these are issues that I can't talk about really at this point. So what was our first key observation in when we evaluated mitochondrial activity in human sperm samples? Well, this is one of the first works we did when I came back to Portugal from my postdoc in the States. And we looked at the human sperm mitochondrial activity from donors, men of proven fertility, from non-zoospermic patients, so patients who look normal, but they're in the clinic for assisted reproduction purpose, so they can't be normal, and for patients who are also in the clinic, but whose sperm is clearly not normal. They have both low amounts of sperm, low motility, and bad morphology. So those are the OATs, those are the worst possible samples you can get. And what we found is that we had clearly in these different samples, the donors had much higher mitochondrial activity than the normal zoospermic patients, and the OATs were by far the worst. So, okay, mitochondria has something to do with this. And well, that was that was the key group that we had. It was the control group, because usually in the clinic, you only get groups B and C, because fertile people don't go to an assisted reproduction clinic. So we had to recruit them elsewhere. But all samples, and that's all cell samples, even your clonal cancer lines, they're heterogeneous. Okay, each cell is a cell. So what we're measuring was populational effects, and we noticed from using both flow cytometry and fluorescence microscopy that some cells had high mitochondrial activity and some cells really didn't. So there, of the three, only one does. And so, can we separate them? So we called people at the cell sorting facility at the hospital. They do that mostly for cancer patients, for leukemia patients actually. So. And we separated them out, and then we analyzed them, and we found that patients with, individual sperm with higher mitochondrial activity we could separate them from those of low mitochondrial activity. But I mean, how do you prove that one with higher mitochondrial activity is better? Well, that was what reviewer one of this paper asked. He said, well, you have to do the experiment. You have to have human oocytes, fertilize them, and show that your sperm with higher mitochondrial activity are more efficient. But of course, you can't do that because well, of course, no, because the reviewer didn't know that. You don't have human oocytes to do experiments on. At least we don't. So what we had to do was to go back to other analysis that I had learned doing my postdoc. One was chemical decondensation. So we used this sort of assay that shows that the sperm participating in a mock fertilization event. And then we used also an heterologous system, which was injecting sperm into an oocyte cytoplasm, but a bovine oocyte cytoplasm. This is cow oocytes, not human. And so what we found was that the sperm with higher mitochondrial activity did the top thing, which is decondense, and so show that it was ready to participate in fertilization, hopefully, while the sperm with low mitochondrial activity just stayed sperm-like. That's the bottom oocyte there, just stayed quiet and really didn't do anything at all. And so that's how we answered reviewer number one. I hate him, and I wish I could find him. Anyway, never mind. Moving forward, so why? Why is this? Oops. Here we go. Another thing we found, which the clinician asked us to do, okay, can you show me that this is relevant for assisted reproduction? And not with this is a different project, because the clinician was interested in ROS and reactive oxygen species. So we showed the clinician that actually you could select better patients for the best patients for in vitro fertilization were those with low production of ROS, using MitoSox ROS, a superoxide probe, which actually also reacts with hydrogen peroxide. And the men with higher ROS production were actually you're better off using ICSI with them immediately, so intracytoplasmic sperm injection. So the clinician was very happy with this, and this is what started to be done in the lab, in the clinical lab. But being biologists, we're really interested in complicated things, right? So, what kind of molecular things are involved? I mean, we were looking at some very basic phenomenological aspects that you could monitor, because sperm viability is also limited in humans. You cannot use it like a cell line. So what can you find here? These are all the metabolic pathways in cells. It's a slide I usually give to my biochemistry students to frighten them a little bit and say you have to memorize all this by the end of the semester, which is not true, by the way. So since we're interested in the mitochondria and metabolism in sperm, we first addressed this with sperm tail proteomics. So to purify our sample a little bit more, we separated sperm heads and tails, and we did proteomics on the sperm tails. This was the first work done by Joao Ramalho, currently in the Max Planck in Berlin. We do not use this technique anymore. This is the first slide we have on this. We now use the fluorescent plex probes for proteomics, but this is the first work we did on this. So what did Alexandra do? Basically, what she did was she was interested in issues related to metabolism, and an interesting readout for sperm, obviously, is motility, right? So here, there are two comparisons. In A, you can see a proteomic comparison of men who are either normal zoospermic, so their samples are normal, and men who are stenozoospermic. That means their samples have the right amount of sperm, they have the right morphology of sperm, but they're less motile than the normal zoospermic patients. So that's a comparison between patients, okay? Different patients. So what you have in B is a comparison between sperm subpopulations from the same patients. So this is from the same patient, you take the sperm that's moving and the sperm that's not moving, and you compare them. So inter-samples, inter-subjects comparison, and inter-sample comparison. As you can see here from the colors, they're very similar. And in fact, what Alexandra found, and this has been published a while ago already, is that the same sort of metabolic pathway She has downregulations of sperm proteins involved in sperm motility, known to be involved in sperm motility and capacitation, and gamete competence and binding and other things. Importantly, and more interesting for me at least, Sandra was not involved, although they have the same last name, they're not related. Sandra also found that the fatty acids metabolism was much lower, was affected in these patients with idiopathic male infertility. So sperm that are somewhat moving, but they're not really working properly. And we're following up on this. And this is the most recent data that we have on these sorts of issues. Just to let you know, I mean, we always get asked, why is this an important topic? So the quality of sperm in many countries when you do sperm, and now this is mostly done in the military when people go in a military-related context for the recruiting process, for example, in Switzerland, Israel, and in other countries, it's seen that the quality of sperm is decreasing. This maybe has no importance towards fertility issues, but it points to another aspect related to overall health of, and so there are many people proposing that this could be kind of a canary in the coal mine for for hidden health issues that people may have. So that's what Renata here is interested in. She is interested in what is called the exposome, because it is thought that this decrease in sperm quality overall involves endocrine disruptions and things in the environment. And what Renata has been showing for a few years now is that small amounts of things, of pollutants, mixtures, and all sorts of other things can affect sperm function, change their calcium signaling, change their capacitation status, change their acrosome change their function, really. And that's another thing that we've been interested in. Funny enough, when Renata published her work, we were contacted by a pharmaceutical company interested in using her data, her assays to test spermicides, which I thought was interesting. We were trying to make the sperm work, and the company was trying to make them not work. Finally, as the final part of my talk, I would just shortly, I would like to mention that I also do comics. So that's my nerd function, I guess. And so this is my comics career in one slide. That is my shop down there. Dr. Cartoon. It's very professional, as you can see. And so I was always interested in cartoons. And this is a message mostly to younger people. If you're interested in something, I have a student who's interested in music, for example, and she's now doing cognition, a PhD involving cognition and music and stuff like that. So this is my hobby, really. So a few years ago, when we had a project on science communication, a colleague of mine said, well, it's a project on stem cells, and you work on stem cells. Would you like to do a comic to explain how stem cells work? And I said, sure, why not? It's something different. It's my hobby. I mean, I don't really do it professionally, but yeah, why not? So this is the comic we did on stem. It explains how stem cells work, really, and what they can be used for, what the different properties are, et cetera, et cetera, et cetera. Oh, by the way, all these materials are available on our website. Funny enough, as soon as I did this, the MD, which is responsible for the hospital we work at, said, well, we should do this for infertility patients to help them figure out, to explain what exactly we do and what we can't do and what are the things that are involved in the treatments and da-da-da. So these are some of the patients, some of the illustrations we did for patients with the Society for Reproductive Medicine in Portugal to explain different aspects related to infertility and how it can be overcome and what can be done. This was also about gamete donation, surrogacy, and other sorts of issues that are differently. The colors in the European map there mean that not all the science you can do is doable in different countries. So in some places it's illegal to do some things. And so it's also important for people to realize that. Well, this was very interesting, and I thought I thought it was cool to be talking to people and trying to make them understand what we do as scientists and as clinicians in this case. But my concern was, as a scientist, was, do people learn anything? I mean, is this useful? I mean, it looks nice, but do people understand the concepts behind this? Do people appreciate what is involved? And so this is a project that we did with science communication officers in our institute. So basically, there's, by the way, there's the spermatogonia and the oogonia down there, the stem cells of the testis and the ovary. So what Sara Varela-Amaral, another Amaral, none of them are related, did was, she is a psychology student, science communication, interested in science communication. So what she did was she ask people questions about infertility, about assisted reproduction techniques, about stem cells, about different sorts of topics. And then she showed them the comics and saw what they learned. And she quantified this. And now she has a career doing this. That's what she does. She used to be a biochemist, and now she does this for a living, which I think is extremely interesting. This is so interesting that when we published this paper, we immediately got called by colleagues who work on metabolism. Had a European project called Foie Gras. There are several of them. This is ours. It's an Horizon 2020 project. And I thought I was going to be called to do metabolomic studies, or use the seahorse or something like that, or look at the sperm or oocytes or ovarian tissue from patients. And they said, no, we want you to do the communication part of the project, which I thought was extremely interesting. The countries involved are those you see there. And so we talked to patients, we interviewed patients, we found out what they knew about their disease. We made a sort of comic that links their life to the different aspects of the disease. And we also asked them what they wanted to know. So a lot of patients didn't, for example, this is just one example. Patients were very concerned about being obese and having a lot of fat and having a fatty liver. But a lot of them said, but I don't eat fats, I avoid them. Yes, but we found out that they ate a lot of sugar, and sugar can be turned into fat, which we know, but the patients didn't know. So we made small points joining the biochemical aspects, because if you only give them biochemical aspects, it won't work. But we joined that with lifestyle sort of things, and it worked, and the people actually understood their condition better. This comic is available in every language, well, not every language, in a lot of languages, including Spanish, in that website right there. It's freely available. It's in all the languages of the consortium, basically. And it led to the publication, the student actually did this in Spanish. That's her PhD thesis. And those are the papers she has, both on how she made the comic and how she evaluated the comic. So yeah, so that's another thing that we do now in the lab. And we have a subgroup. I have subgroups for many things. The subgroup on sperm metabolism is Sandra Amaral. On oocytes, it's Ana Paula Souza. And here in the communication, it's Marisa Azul, who collaborated with me on this. Anyway, to do all this, you need people to help out. So we have people in the sociology department and the psychology department, which we need to interact with patients. We have people at the hospital for the flow cytometry and cell sorting facilities. We have people with that gives us access to patients in different hospitals. The proteomics is done in collaboration with Rafael Oliva at the University of Barcelona in Spain. All the other aspects are done, the female aspects of this are done in collaboration with Teresa Woodruff in Northwestern in the US. The patch clamping of sperm is done with Chris Barrett at Dundee and Steve Public over in Birmingham. All the testicular work is done with Stephen Schlatt, and the stem cell work is also done with my brother at the University of California in San Francisco, where I was for a while. Okay, and this is where I am. The black dot there on the left is where my city is. That's my city over there. Very old. The university was founded in 1290, as you can see on the top of the slides. And here is the group that did all the work, so I can talk about it. As you can see, well, we have veterinarians, MDs, psychologists, engineers, communicators, journalists, you name it, we have it. What we don't have is gender diversity, as you can clearly see here. And that is something that I am a bit concerned about. But anyway, it's what it is. So that is what I had planned. I think it was half an hour, so that was basically it. So yay. I'll take any questions that you might have or not.
Well, thank you very much, Dr. Ramalho-Santos, for your very interesting lecture, especially the last part of your work in outreach. I will found very interesting. I will open the discussion for the audience. Any question for them? Ingrid? Ahí hay micrófono.
Joao, can you see me? Yes, here.
Yes. Hi. Hi.
Very nice presentation. I am an electrophysiologist, so I would like to know which channel are you going to, so in what channels, or
Okay.
what is your question related with ion channels in sperm? Thank
Sure.
you.
No worries. So actually, what we're looking at is the ORI channels, the ORI calcium channels. Okay. So this is a project by Inés Alfayat. I'm hoping she has some data by the time I turn off this talk, because she's currently now in Dundee. And so her work has to do with, since these channels are modulated by fatty acids, right, and other sorts of things. So she is interested. She has a basic biology part of her project, which is modulation of these channels' activity using patch clamp. And she also has part of a clinical part working with obese patients to see if that influences the sperm function. Okay, so she has two things: with normal sperm, adding stuff to sperm, and then using sperm from obese patients. So those are the two points of her thesis. But she has not. She has patch clamped cat spur, which is not really a news, right? And now she's trying to patch clamp these. This has not been done yet that I know of. Okay, in sperm.
Thank you.
Sure.
But do you have any data that actually ORI is working in the sperm? Oh, not yet.
We have not with patch clamp.
All right.
Okay.
Yeah.
Not with patch clamp. No, that's we want to have data with patch clamp. Although if I had, I would show you, because that would be very nice. What we have is the usual. You throw inhibitors at stuff at sperm and see what happens in terms of downstream events. So capacitation, acrosome reaction. So these kinds of downstream events of sperm function. But we have not monitored the channels directly. And the point with the inhibitors is that, as you probably know, they are very unspecific and they can do a lot of different things. So unless you're patch clamping the exact current and you can show some information there, I'm not going to be comfortable with it. So she did all the preliminary things. She found the channel in sperm, which is important. She found all the subunits with Western blot immunocytochemistry. She did inhibitor experiments showing that they can cause issues to sperm function, but now she's going to more the channels themselves. I hope that answers your question.
Thank you very much.
Thank you very much.
Nope.
Dr. Garrett Owen is going to ask. Yeah.
I can't hear anything. Am I supposed to?
Okay, can you hear me now?
Sure.
Wonderful.
Yes.
Okay.
Well, thank you. A lovely talk. And I'm actually going to apologize for breaking the rule about asking questions. I'm going to ask you a question that is not related to anything you actually presented in your talk. But I'm going to take advantage that you're very well known in this area to ask a question that I've never had the answer to. We know that when there's ovulation, that when the oocyte comes out, it comes out with all the fluid that's in the anthro. And there's hormones, there's buffers, there's glycoproteins, lots of different information. And we also know that when the sperm finally makes its way out of the body, it has got all the juices that come from the seminal vesicle and from the prostate. But I also know, because I did my PhD in an infertility clinic, is that when you do in vitro fertilization, what you do is you take all those substances away and you just add the sperm and the egg together. So I see you're actually looking at motility and metabolism. And I'm just wondering, how are you looking at it? Are you looking at it in the actual in vivo context where all these factors from the fluid are present? Or are you looking at it in an isolated way that's in vitro fertilization?
Excellent question. Well, it's a question that it's not easy to get to the. It's not the answer to your question, okay? But it's a reason why we often can't get to the answer you're trying to the answer you want. Is that whenever we have these problems, people just inject the sperm into the oocyte and don't forget about the rest, right? And so, for example, in the Middle Eastern countries, the intercytoplasmic sperm injection is basically 100% the technique used, and nobody does in vitro fertilization that that Patrick and Bob did, and that won them when Bob the Nobel Prize because Patrick was deceased. So nobody does that. That is a very interesting question. We do that. We cannot do motility we cannot do the experiments with the fluids. That is just not possible. What we're trying to do is also do the proteomic characterization of the fluids, both male and female. And we have now a student that is working on the oocyte fluids, and she's trying to create more or less what you might call a female oocyte-related thing. But again, the issue here is more complicated because, the sperm comes out with fluids, right? But then it goes into the female reproductive tract, where it loses some proteins, gains some proteins. There's great Japanese groups doing in vivo following of the sperm through the reproductive tract. But the logical answer to your question is I have no idea. Even the experiments that show that some ovarian in human, that some ovarian follicle substances, namely progesterone, might serve as chemoattractants, for example, the evidence is not that great, if you ask me. So I know that's not the answer to your question, but I don't have it. So you'll have to keep looking. Sorry.
Okay, thank you very much.
No worries.
Thank you very much. And any other question? I was wondering, Dr. Ramalho-Santos, specifically, I'm thinking in intracytoplasmatic sperm injection, and there is any evidence, hard evidence, related to the impaired metabolic function of the sperm and genomic integrity or genomic function of the sperm?
In intracytoplasmic sperm injection. There is some studies, if you're interested, the literature to look for is Rita Vacena's group in Barcelona. They're doing, because they're a human clinic who actually can use the unfertilized and sometimes even fertilized embryos to do proteomics experiments and other sorts of things. There are very few groups that can do this. The use of ICSI, basically with ICSI, you get 100% fertilization, right, by definition of a sperm being inside an oocyte, because you put it there. So a lot of people aren't interested in looking further than that. What I can tell you is that the success rates for ICSI in terms of embryo development are not that great in most places, which has to be due not to fertilization, quote unquote, but to something else. What that something else is, we don't know, okay? The people are looking at different sorts of mutations. There's a European consortium looking at histone mutations and other sorts of mutations in sperm, in the male gamete. But the answer is we really don't know about that. And it's very hard to think about experiments where you can address this using the proper controls, because the proper controls are often embryos. I mean, viable embryos. And at least here, it's very hard to use those. Okay.
Okay. Well, thank you very much, Dr. Ramalho-Santos, again. And we are continuing with our symposium. Thank you very much.
Here, bye-bye. Have fun.
Bueno, vamos a continuar con nuestro simposio. El siguiente expositor es un conocido amigo nuestro, doctor Ricardo Moreno. No sé dónde está. Ajá. No te iba a presentar. No. Sí, no necesita presentación. Nuestro amigo Ricardo Moreno es doctor en ciencias biológicas con postdoc en Estados Unidos. Él dirige su laboratorio relacionado con la espermatogénesis y la función espermática desde hace mucho tiempo en la Pontificia Universidad Católica de Chile, en el Departamento de Fisiología, en la Facultad de Ciencias Biológicas. Y para nosotros es un placer escucharte de nuevo, Ricardo, y el título de su presentación es Modern Lifestyle and Sperm Function Lessons from the Mice Models. Muchas gracias, Ricardo. No sé, pero aquí me pusieron tu título en inglés.
Gracias. Como dijo, gracias. Sé que como dijo que, ¿no es cierto?, acá Enrique, en el departamento de fisiología, es que de lo poco, o sea, que de lo poco, ¿no es cierto?, es que queda. Hace unos 5 años éramos 15 profesores, hoy día quedamos 6, y no sé cómo queda. Entonces, hoy día yo les voy a contar una historia que está algo, yo creo que algo ya que Joao dijo. Les voy a contar cómo el ambiente del siglo XXI o del siglo XX podría impactar en forma negativa a la función del espermatozoide. Y la historia comienza ahí con 2 mujeres. Rachel Carson, en el año 1962, publica un libro llamado Silent Spring. Ella parte el libro con un cuento diciendo que cuando chica iba al campo, no sé qué estado era, Utah, que Wyoming, algo así, y se fue la diapositiva. Bueno, y lo que ella cuenta es que habían pastos, habían árboles, hojitas, iban al lago, pescaban, súper bonito. Pero con el correr del tiempo y los años, en el lago habían cada vez menos peces, habían menos pájaros, habían menos flores, hasta que al final todo se desvanece. Y en los próximos capítulos presenta evidencia de cómo los pesticidas y los herbicidas matan no solamente a las plantas, pestes y los insectos, sino que quedan en los ríos, en los suelos, y matan a todo el resto de la fauna y la flora, insectos. Después de este libro, la policía, el Senado, la toma a ella y la investiga, pensando que era una comunista en contra de US. Estamos en los mejores tiempos, recuerden, de la Guerra Fría. Bueno, ella sale indemne. Y la segunda mujer es Theo Corbin, que era una química farmacéutica, dueña de casa después, se casó, tuvo hijos, tuvo nietos, y después cuando tuvo nietos dijo, voy a estudiar. a los 58 años saca su doctorado e investiga de nuevo el efecto de los contaminantes ambientales sobre la fauna silvestre. Y lo que ella encuentra es que estos contaminantes en la fauna silvestre afectan el desarrollo del embrión, o sea, una etapa crítica. Con estos datos, convoca a un congreso. En el año 1991, ella dice, estos compuestos los llamamos perturbadores endocrinos. Ella es la fundadora del término. 58 años recién que su doctorado, o sea que nunca es tarde para esto. ¿Qué son? Es un agente o una mezcla de agentes que interfieren con cualquier aspecto de la función de una hormona, ¿ya? En la síntesis, en la unión, en su receptor, órgano, etc. ¿Dónde están? Están en todas partes. Pero hoy día que nos vamos a ver en los plásticos. En los plásticos están en todas partes, en los Tupperware, en los computadores, en este micrófono, y están compuestos de dos tipos de moléculas. Uno que son los alquilfenoles, que hacen polímeros, o sea, pero son duros. Y la industria entonces que para que sean blandos, flexibles, les ponen unos compuestos que son los talatos. ¿En qué es ese olor a plástico aquí que ustedes encuentran? Quiere decir que los talatos, como son volátiles y se unen en forma covalente al plástico, se vuelan. Y ustedes los huelen. Y son liposolubles. Y ustedes entonces los absorben. ¿Ya? Entonces están en todas partes. En la vía, en las latas de conserva, botellas, etc. Y los papers dicen que estos compuestos se han encontrado en líquido seminal, orina, leche materna, lágrimas e incluso en el líquido amniótico. Joao mencionó el exposoma. 85.000 son las moléculas o partículas que se estima que nosotros estamos expuestos desde la gestación hasta que somos adultos. El smog y la contaminación y los plásticos y qué sé yo. Entonces, alguna de esas a lo mejor simplemente no es cierto, es que no hace nada. Y otras que sí. ¿Cuáles son? Entonces, el inconveniente de esto es cómo se estudia. ¿Cuáles son malas? O sea, que cuáles son buenas. O sea, que cómo que se estudia, ¿es un efecto crónico o un efecto agudo? Lo otro, ¿qué compuesto? No es como que ustedes quieren ver, ¿no es cierto?, acá una droga, o sea, que hay una pastilla y esta es la única fuente. Acá están en todas partes. Entonces, ¿cuál es la dosis a que yo estoy expuesto? ¿1 miligramo? ¿100 miligramos? ¿Ninguno? Y lo otro es, si sea 85.000, ¿cuáles voy a estudiar? ¿Cuál va a ser, no es cierto, que la concentración aquí la de cada una? Entonces ustedes, si ven papers, que van a encontrar miles de papers, hay gente que ve un compuesto, que ve dos compuestos, que ve 4, 20, etc. ¿Ya? Y lo otro depende de lo que uno esté expuesto. Hay, por ejemplo, así como dos tipos de formas. Uno que se llama una exposición ocupacional. Es decir, gente, ¿no es cierto?, que, por ejemplo, que trabaja en las fábricas de pintura. Están expuestos constantemente. Y nosotros, exposición no ocupacional, en que estamos expuestos, pero varias cosas. ¿Ya? En los modelos, ¿cómo se lo dan? Usted que tiene, ¿no es cierto?, acá una rata y un ratón. O sea, ¿cómo se lo damos? En crema, que lo aspiren, que lo tomen, o sea, que se lo inyecten. ¿Y qué es lo que podemos medir? El peso, o sea, que cómo se comportan las hormonas, órganos, etc. Y lo que nos gusta, o sea, que como dijo Joao, ¿qué es lo que hacen? Mecanismo, ¿ya? Pese a esto, ¿no es cierto? Hace como 5 años, con un grupo de 4 estudiantes en el doctorado, pensamos y dijimos, ¿sabe qué más? Si que hagamos algo. Tomemos 5 compuestos en los plásticos, ¿ya? 2 alquilfenoles, el NP, en que está en los detergentes, en polvo, el octilfenol, que está en las pinturas, el BBP, que está en los pegamentos, o sea, de ejemplo, en la cola fría, el DBP, que están en los cosméticos, y que por esto es una causa importante de las niñas puber, ¿no es cierto?, que cuando que se empiezan a, o sea, que cuando que se empiezan o sea, que, ¿no es cierto?, que a maquillar, estos compuestos pueden, y hay algunos datos, ¿no es cierto?, o sea, que le cambian sus hormonas, por ejemplo. Y el DHP, que está en las bolsas de plástico. Un compuesto que a lo mejor ustedes conocen y que no usamos es el bisfenol A, EPA, que hace años hubo tantos datos, que este compuesto en humanos induce cáncer de mama, que ustedes ahora van al Jumbo, ¿no es cierto?, se ven marcas de botella sin este compuesto. Pero es el bisfenol A. Si ustedes le ponen por ejemplo, una molécula de cloro, es el bisfenol C. Y ese es otro compuesto. Y si le ponen otro, que tenemos el D. Y ahora tenemos 5 tipos. En que básicamente a lo mejor hacen lo mismo. ¿Ya? Entonces, lo pusimos en 2 mezclas. Una mezcla alta, pensando en la mezcla ocupacional, o baja, no ocupacional, y se lo dimos en el agua a las hembras de ratón durante toda la gestación, durante su lactancia, para que pase vía su leche materna a las crías. Separamos machos y hembras hasta que tuvieran una edad de 90 días de edad. En los machos, ¿qué fue lo que vimos? Una disminución en los niveles del estradiol y una disminución en la razón del estradiol a la testosterona. Además, vimos cambios en los niveles de los mensajeros de varias enzimas de la vía esteroidogénica, por ejemplo, no sé, CYP11A o la CYP19A1. Además, los machos y no las hembras, aumentaron su peso, bajó el peso de los testículos, bajó el diámetro de los túbulos seminíferos, bajó la altura del epitelio y el área de los túbulos, y cambios en la frecuencia de los estadios de la espermatogénesis. Hasta ahora, entonces, que la conclusión es que la exposición crónica de una mezcla definida de los ftalatos y alquilfenoles en dosis equivalentes a los niveles de la exposición en humanos tiene efectos de un perturbador endocrino en los ratones machos. O sea, funciona el modelo. Funciona como un perturbador endocrino. ¿Qué pasa con los espermatozoides? Encontramos que hubo una baja en los espermatozoides de la cola del epidídimo, o sea que en la zona donde están maduros. Y desde un punto de vista funcional, medimos la reacción del acrosoma, que es cuando esta vesícula que se encuentra sobre el núcleo vierte su contenido hacia afuera y que puede pasar en forma espontánea, in vitro, como acá, ¿no es cierto? O bien se puede inducir por un supuesto inductor fisiológico como es la progesterona. In vitro, entonces, nosotros vimos que la reacción espontánea fue igual entre los espermatozoides de los ratones expuestos solamente a los vehículos y aquellos expuestos a la dosis más baja, ¿ya? Pero los espermatozoides de los ratones expuestos, fíjense, fue mucho mayor que los de vehículos. Además, in vitro vimos que hubo una disminución en la fecundación solamente con la mezcla menor. Entonces, y in vivo, además, no tenemos datos de los machos, pero se los voy a mostrar en las hembras, solamente este dato. Las hembras, cuando las pusimos con los machos o expuestos, vimos que hubo una baja en el largo de la gestación, que las crías tuvieron un bajo peso al nacer, y estas crías, de nuevo, crecieron adultas, y las pusimos con machos, de nuevo, F2, también un bajo peso al nacer, viabilidad de los fetos menor y una disminución en la distancia anogenital. Esto es lo que se llama un efecto intergeneracional. Es decir, si a mí que no es cierto es que se me expone algo, sería una F0. Mis hijos tienen un efecto, F1. Y mis nietos también que tienen un efecto, F2. Pero ninguno de ellos dos tuvo una exposición. Esto pasa y que, por ejemplo, un paper ellos muestran aquí, que es lo que mencionaban en la mañana, a los ratones machos F0 se les dio una dieta de cafetería, ¿ya? Y luego se los pusieron con hembras o expuestas y vieron la F1, F2, F3 y F4. Y lo que ellos vieron, desde que la F1 y la F2, tanto en machos como en hembra, hubo un aumento del peso, ¿ya? Se eronó en la F3 y 4. De manera súper interesante, lo que ellos vieron ahora, aislaron el RNA de los espermatozoides. Y lo que vieron, este es el patrón, es decir, el tamaño de todos los RNA que lleva los espermatozoides. Controles y los de la dieta. Vieron, oh sorpresa, hay un cambio, ¿ya? Entonces, nosotros pensamos que a lo mejor es que también había esto. Y lo que vimos es que en los espermatozoides de los ratones expuestos encontramos un aumento en los RNA ¿Ya? Y además también encontramos un cambio en el perfil. Acá vemos el perfil de los espermatozoides de los ratones expuestos al vehículo, y esto es de los espermatozoides de los ratones expuestos a la dosis más baja de 1 miligramo por ml. Después de hacer una secuenciación masiva, vimos que acá le ponen genios, pero no son genios en realidad, son tipos tipos totales de los RNA, un cambio en 1/3 de todos ellos, ya sea aumentaron o bajaron, ¿ya? Y hay de varios tipos: mensajero, microRNA, RNA pequeño, RNA largo, RNA mitocondriales y ribosomales. De todos estos, que los que cambiaron, 2/3 aumentan, 1/3 baja, ¿ya? Y lo interesante es que de todos los que cambian, todos los ribosomales bajaron sus niveles. Y los mitocondriales, todos aumentaron. El resto cambió, ¿ya? Y esto, ¿no es cierto? Y de estos tienen varias funciones. Por ejemplo, en la espermatogénesis, roles en la inmunidad y en el metabolismo de los lípidos y del azúcar. ¿Ya? Y que por el tiempo, ¿no es cierto?, es que no les cuento más. Entonces, la conclusión es que este podría ser un buen modelo de una exposición crónica, es decir, desde la gestación hasta que se es adulto, y que tenemos un efecto no solamente en las hormonas, sino que también en la función del espermatozoide, y esto que a lo mejor es que podría tener un impacto al menos en el desarrollo temprano, ¿ya? Y que es posible que además en el sitio de la fecundación también tenga un impacto debido a una exposición directa, ya que también es que se han encontrado en el líquido folicular. Lo último, dos al menos consideraciones finales, es que entonces hay que cambiar el paradigma de lo que es un espermatozoide. Estamos pensando, ¿no es cierto?, que el espermatozoide lleva solamente el núcleo. No es así. Hace más de 100 años que sabemos que también aporta el centrosoma. Y ahora estamos sabiendo que además aporta los RNA. Entonces es mucho más que un núcleo, ¿ya? Y lo segundo, dado que si uno se ve expuesto a algo y su F1 y la F2 obtienen el mismo cambio, sin un cambio genético, sin una mutación, esto es lo mismo, esto es súper antiguo, tiene más de 100 años. Lamarck, antes de Darwin, pensaba que esa era la manera de cómo se cambian las especies. La jirafa, ¿no es cierto?, tenía el cuello corto, las hojas, ¿no es cierto?, sé que las come, están arriba, estira el cuello. Y sus hijos tienen el cuello más largo. ¿Por qué? Porque siguen alcanzando, hasta que los nietos alcanzaron. Llegó Darwin con su libro, que es Selección Natural de la Especie. El pobre Lamarck es que lo patearon y lo dejaron acá en el olvido. No sirve. Pero ahora la gente se está dando cuenta que es importante, ¿ya? Eso. Ya está, o sea, esta es la gente aquí que me ha ¿no es cierto?, ayudado aquí en este camino, y gracias.
Muchas gracias, Ricardo, por tu exposición y abro la discusión a la audiencia. Preguntas, por favor, adelante.
Pregunte, si no, está bien. Si no, quiere decir es que no entendieron nada o si lo entendieron todo.
Gracias.
Profesor. Para mí siempre será profesor, así que. Primero voy a defender a Darwin. Yo creo que los fanáticos son siempre los peores. No, Darwin en el libro lo que dice, en un capítulo establece que la principal ley de la evolución es la ley de las condiciones de existencia. Ahora, los que vinieron después, ciertamente borraron todo. Pero pasando a la presentación, respecto de los microRNA, ¿ustedes han mirado en detalle cuáles son? Y lo pregunto por lo siguiente, porque hay un estudio muy interesante sobre abuso sexual en machos que muestra, primero, lo que ocuparon ellos, hicieron una expresión diferencial en hombres, ¿ya? Identificaron microRNAs que se expresaban de manera distinta a nivel de espermio. ¿Por qué? Porque muchas veces los hijos de hombres abusados sexualmente durante la infancia tienen episodios de psicosis y alteraciones en otras conductuales cuando son adolescentes. ¿Ya? O sea, el padre estuvo expuesto a algo y el hijo sufre una consecuencia. Y luego lo probaron en un modelo de estrés social en ratones. Identificaron cuáles eran los microRNAs que también se alteraban en los espermios de los ratones y luego los modificaron. Y lo que demostraron es que se podía revertir este fenotipo heredado. Entonces, ¿ustedes han pensado en mirar así cuáles son los microRNAs que están alterados ahí? No va a ser el mismo resultado, sino que va pensando en cuáles son las maneras de revertir esto.
Mira, cuando estaba haciendo esta presentación dije, capaz que haya alguien que me pregunte eso. No nos falta, nos falta el que pregunta lo que uno dice, no, ¿qué va a hacer? Y no me cambia acá. Ahí está. ¿Viste? Y dije, no nos va a faltar que me pregunte, ¿y qué pasa con los microRNA? Bueno, esto es una lista de los que aumentan acá, ¿cierto? Y de los que bajan. De los que aumentan están en rojo y los que bajan están en azul, negro. Y les voy a marcar, estos son los micro, ¿no es cierto?, que RNA en que nosotros encontramos y que también que se han encontrado en varios estudios, ese que ¿no es cierto?, el que tú dices, o en otro en que, ¿no es cierto?, que separan a las crías, los de la madre, o sea, en el estete, y las separan por 1 hora, por 5 o 6 días. Y ese es un modelo también, ¿no es cierto?, aquí que es 3. Entonces la idea es que hay componentes que son importantes, transversales. Hay un paper en el NAS de hace como 10 años en que mostraron que el 34C, si no está presente al instante de la fecundación, en el ovocito no se activa. Ya, o sea que pareciera que todas las condiciones, la que uno que no es cierto aquí que se expone, tienen componentes similares. Eso sí que los vimos, gracias. ¿Viste? Sí, yo sabía que alguien me había preguntado.
Dolores y Miguel ya. No, Dolores primero y después Miguel. En eso.
Ricardo, gracias por tu presentación. Yo te voy a preguntar si analizaron algo más allá de lo reproductivo. La parte metabólica me parece interesante obviamente por la función que tienen las hormonas en regular, por ejemplo, los lípidos y el metabolismo de la glucosa. ¿Miraron algo de eso o todavía no? ¿O piensan hacerlo?
Y fue súper interesante porque era un estudiante que estaba haciendo el doctorado de estadística. Nunca había entrado en su laboratorio en toda su vida. Nunca había tomado una micropipeta, nada. Y es lo mejor que les puede pasar porque esa gente que no está contaminada con nada, entonces uno llega y que le enseña como uno quiere y hace las cosas como uno quiere. Súper bien. Y él, ¿no es cierto?, es que miró eso. Y hubo un aumento de la grasa parda, me acuerdo, disminución en leptina, él vio, y un aumento en los adipocitos. Algo así es lo que me acuerdo. Sí, o sea, pero eso que lo miramos. Sí.
Gracias.
Miguel.
No, y que además que
Gracias. Gracias,
vimos
Rodrigo.
que, ¿no es cierto?, que un aumento en el peso solamente de los machos y nos preocupamos que ninguno que de los dos grupos coma más, además. Así que esto que parece que también son obesogénicos, que le llaman. Dale.
Hola. Te voy a preguntar lo siguiente. Mira, en este caso de los cambios con microRNAs, ¿cierto? Fundamentalmente, que es una cuestión re importante, al parecer, digamos, todo el tema de problemas, como tú lo planteaste, en F1, F2, F3, etcétera, etcétera, está asociado a muchos microRNA. Y tú has visto, yo algo vi, no sé si en estrés o algo por el estilo, alguna vez algún microRNA que esté asociado a cambios en el metabolismo de la glucosa, particularmente en aumento de gluconeogénesis a nivel del hígado. Es bien específico, pero es una cuestión re importante porque de alguna manera también te causa problemas con resistencia a insulina y todo ese tipo de cosas.
Que yo recuerde, nunca lo hemos mirado, o sea, pero debe haber, ahí están que los datos, cosas
Seguramente,
que es de tomar
sí.
es que la base de la de datos empezar a ver de nuevo, o sea, pero nunca que lo hemos visto.
Ya.
Gracias.
Y otra cosa que te quería preguntar, que es súper interesante todo el tema de los disruptores endocrinos. Yo lo encuentro que es bien increíble los resultados que se obtienen ahí. Fíjate que hace mucho más de 15 años, cuando yo todavía trabajaba en espermatozoides, nosotros con Walter Sierra Alta hicimos unas tensiones de oro y encontramos unos aryl receptors que no tienen por qué estar en el espermatozoide. Entonces, ¿tú crees que algunos de estos disruptores endocrinos de alguna manera estén incidiendo en la síntesis de algunos receptores que no están asociados a ninguna cosa fisiológica, por decirlo así, que de alguna manera tienen consecuencias que son bastante deleterias, digamos, para la función del espermatozoide?
Gracias, sí. Lo más probable, porque estos perturbadores, sobre todo en la, ¿no es cierto?, que exposición crónica, van a tener un efecto en la expresión génica de toda la maquinaria durante la espermatogénesis. Y eso puede cambiar los niveles que de los componentes y de cómo que, ¿no es cierto?, que se comporte o sea, esta célula. Así que sí, o sea, que de todas maneras pueden ser esto. Y además que pensando en tu pregunta, recordemos que la secuenciación, la del genoma humano, pensamos que iba a encontrar todos los genes y la sorpresa, que al menos que de la manera de cómo el gen se define, fue de un 10%, un 5%, y todo el resto, sorpresa, son elementos reguladores. Así que más importante que si los genes aumentan o no, los genes mismos, parece que es cómo se regulan. Y entre ellos están los microRNA. O sea, que de nuevo el punto, cómo este cambio, que no es genético, pasa para tu hijo y para tus nietos. ¿Qué hace que se mantenga este RNA?
Exacto.
Eso,
Tenemos tiempo para una última pregunta, Alejandro.
Gracias, Ricardo, por tu presentación.
ahí está.
Muy interesante. Y ya me queda claro que el hecho de que un envase plástico diga 100 BPA es un placebo porque puede tener otro plastificante que puede ser igual o peor. Yo te iba a preguntar respecto a los microplásticos, que se habla mucho respecto de la presencia de microplásticos en fauna marina, en sangre y en otros tejidos, y si se sabe qué hacen los microplásticos en el eje reproductivo, porque tú hablaste de moléculas que están en el plástico. pero me parece que el microplástico es una partícula más grande, ¿cierto?
Sí, sí, en el fondo, cuando los plásticos que nosotros usamos se rompen, ahí se forman estos microplásticos, que son partículas súper pequeñas. Ahora, ¿cómo se rompen? Por ejemplo, hay datos, hay paper, que la gente ha tomado un biberón de plástico y lo lava, o sea, que con un hisopo. Toma esa agua, lo lava de nuevo y lo sigue lavando 3, 4 veces, y las aguas en las que tú lavas se encuentran estos microplásticos y las moléculas. Entonces los microplásticos es una etapa intermedia, en el fondo, entre el plástico y cuando ya la partícula ya no es cierto que se disuelve entera. Ahora, no es que uno no use nunca más plástico, dejáis que no se trata de eso. Lo que pasa es que muchos no nos damos cuenta que los plásticos tienen una vida útil, y de hecho te dicen, no sé, pues las sillas, por ejemplo, en las de auto, para los niños, tienen una vida útil de 5 años. Si usted no la cambia en 5 años, el plástico, cuando hay un choque, se le puede romper. Y no va a ser culpa del plástico, fue porque usted que no leyó. Dejáis las bolsas en las que te dan en el supermercado, la fruta o las papas, no se ponen en el microondas. Hay gente que la pone para calentar. Hay cosas donde uno lleva la comida que solamente que para el freezer, guardar, y la gente la usa para el microondas. Entonces, hay tipos de plástico distintos.
Muchas gracias, Ricardo. Perdón, pero me dicen que nos está esperando una expositora desde Inglaterra que ya está esperando. Así que muchas gracias nuevamente. Te voy a pedir especialmente una botella de plástico. Te entregamos un recuerdo, entonces. Gracias por tu exposición. Muchas gracias, y voy a dejar a Ingrid entonces un aplauso para Ricardo. Y voy a dejar a Ingrid con la presentación de nuestra próxima invitada. Muchas gracias.
Hello, Enrica, can you hear me?
Hi, yes, yeah, I can. Hi, Ingrid.
It's so great to see you.
Yeah, absolutely. Totally agree.
So I will stand here for Enrica to see me because the camera is there. So it is a great pleasure to have Enrica Bianchi today as a speaker. Dr. Bianchi will present her talk called The Essential Role of Cell Surface Proteins in Fertilization and Embryo Transport. Enrica is currently a research fellow at the Biology Department of the University of York in UK. She was also postdoctoral fellow of the Wellcome Trust Sanger Institute in Cambridge with Dr. Gavin Wright. And she holds a PhD in reproduction and development from the University of Rome. And Enrica was widely known for many achievements in science, but actually she and Dr. Wright were the one who found the receptor in eggs called Juno, that is the one who binds isomo in a sperm, who was like a big achievement in the field. So, and then after, she has been working in other cell surface protein interaction and also in embryo transport. So I don't know if I have anything else to say. Just welcome, Enrica, and we are ready to listen to you.
Thank you very much. Thanks a lot for the invitation. It's a pleasure to be here. I'll start sharing my screen. Okay. Can you see my presentation? Like that?
Yes, we can see it.
Can you see it full screen? Is it all right?
Yeah, we see it correctly, right, Christian? Yeah, everything is fine.
Okay, yeah. Right, so thanks a lot for the invitation again. Today, as Ingrid was saying, I'm going to talk about two slightly different subjects. One is fertilization and the other one is embryo transport, but in both cases, there are cell surface proteins involved. Right. Presentation is not going ahead. Okay, right. So I'm sure I don't need to introduce or speak a lot about this schematic drawing here. Don't introduce these gametes to you, but I really wanted to draw your attention to the last three steps in panel B, basically, and which are the steps. And these are the steps that I'm going to talk about today. And I'll try to be specific as possible, distinguishing between sperm egg binding and sperm egg fusion. Binding, I will be referring to binding, indicating that the membrane of sperm and egg are still separated. And then when the two membranes fuse together, I'll be referring specifically to sperm egg fusion, and then I will touch briefly on this on the polyspermy block in mammals specifically. So I think nowadays we have not a clear picture, but a decently populated picture, I would say. So there are many proteins that are that have been discovered recently and that they have been identified as necessary for fertilization in mammals, and not only in mammals, also in fish, some of them. So I think the pictures is becoming more and more interesting, but still this doesn't mean that we have understood the entire mechanism of sperm-egg interaction, and I hope I will convince you of this at the end of my talk. But I wanted to actually start from the beginning. So when we started our work, there was only one sperm protein known to be essential for fertilization, which is isomo-1 here. So that was the only protein that was proven essential for fertilization, and that stand the test of the mouse knockout models. So what we did to try and identify the receptor on the oocyte for Izuma-1 was to take a wide, large screening assay approach, which is what we normally do in the lab. And also we took advantages of the technique that was developed in the lab to specifically study the interaction of cell surface proteins. So we express the extracellular domain of cell surface receptors. We oligomerize them. In this case, it was Izuma-1, the ectodomain, pentamerized. So we have normally 5 molecules per each compound, basically. We use this as a probe to try and find the binding partner. So I screened a large library of a cDNA library obtained from immature mouse oocytes. And I screened the library using Izuma-1 as a probe, and we call it a prey in the lab normally. So when I did the screen, then after a while, I was finally able to identify the single clone that was able to induce positivity in the cells when transfected into the cell, meaning that the plasmid contained the coding sequence for the receptor of Izuma-1. And when we sequenced that plasmid, it came out that the sequence was for a gene called the time folate receptor 4. The gene name was given to this receptor because it has quite a high homology with other members of this receptor family. But there are some differences. And actually, when we tested whether folate receptor 4 was able to bind folic acid like the other members of the family, it turned out that folate receptor 4 doesn't bind folic acid at all in vitro when you test it. So you can see here the binding of folate receptor 1 to folic acid. The same is true for folate receptor 2. And this doesn't happen for folate receptor 4. So we could show that, and we could tell also just by looking at the sequences that there are differences in the residues. These are the amino acids that are involved in the direct binding of the folate receptors to folic acid. And you can see that those amino acids are actually different in folate receptor 4 compared to the others. And these differences, together with the assay that we did in vitro, really convinced us that the molecule wasn't able to bind folic acid. And due to this fact, and to the fact that the gene turned out to be essential for fertilization, we suggested to rename it Juno, after the Roman goddess of marriage and fertility. So what we did then was to test whether the protein was able to was really involved in fertilization, basically. So we added an antibody in order to see whether the antibody was able to prevent fertilization in vitro, and that was exactly the case. So when we added an anti-Juno antibody that was commercially available at the time, we could nicely see that fertilization didn't happen, indicating that binding of the antibody to Juno prevented the binding of Izuma-1 as well. And again, indicating that the protein was involved in fertilization. And then we could confirm those data by generating knockout animals. And the female that do not express Juno, they are completely infertile. The infertility is due to the inability of the eggs to be fertilized. And we wanted to distinguish or try to distinguish between binding and fusion. So we really tried to figure out whether Juno and Izuma interaction was involved in the binding or if they were also sufficient for fusion. So I counted, basically, I counted the acrosome-reacted sperm, which are the sperm that expose Izuma-1 on the surface of their head. I counted those sperms during the in vitro fertilization assay, and I found that those acrosome-reacted sperm are less efficient in binding Juno knockout eggs, indicating again that Juno and Izumo interaction is involved in the binding of sperm and egg. So then we wanted to test whether the two proteins are also sufficient for inducing fusion in cells. So we transfected different populations of cells with Juno and Izumo, mixed them together and checked for fusion events. And we couldn't detect any. So these data altogether, they indicated that the binding pair is essential for fertilization, is essential for the binding of the two gametes, but is not sufficient to induce fusion. And we also confirmed this data with other assays and we never actually published the results, but I think it's finally, well, it's nice that recently there was another paper indicated that also in rat, Izumo 1, for instance, is required for binding of sperm and egg, but it's not sufficient for fusion. After that, we investigated a little bit the role of Juno and Izumo 1 in the cross-species specificity. We know that gametes don't. There are barriers depending on the species from where the gametes come from, and we wanted to test whether these barriers could be due to the inability of Juno and Izumo to bind to one another. So, and we particularly focused on the hamster egg because you might know that, that the hamster is a little bit, the hamster egg is promiscuous in the sense that it can fuse with mouse sperm, with human sperm, and with pig sperm, I think. So we tested whether there was a correspondence, let's say, between the ability of the egg, of the hamster egg, to fuse with those sperm from other species, and the ability of the hamster Juno to bind Izumo from the different species. And this turned out to be the case. So, as I said, hamster egg is able to bind and fuse to human sperm. And as you can see here, the protein Juno from the hamster binds nicely to human Izumo, as does with the mouse homologue of Izumo 1, and also it binds nicely to pig Izumo 1. So indicating that maybe Juno and Izumo are involved in the establishment of cross-species barrier. Again, to test this binding, we used the assay that was developed in the lab, which requires the expression of this oligomerized protein, which in this case was Juno, and then we had Izumo 1 arrayed on plates. And you can easily screen for the interactions with a colorimetric assay. On the other hand, when you test whether mouse Juno binds human egg, then you get a negative result. And this is in agreement with the fact that mouse eggs do not fuse with human sperm. So all this data seems to indicate that the two proteins are really involved in cross-species specificity. So as I was mentioning at the beginning when I showed you the picture, I said that when we started, it was only Izumo 1 that was known to be essential for fertilization on the sperm side. And then a couple of years ago, I think the second protein came out, was identified actually initially in the cattle industry. The protein is called TMM95. It was identified in a genome-wide association study because there was a population of bull used for reproduction, of which a small subpopulation were subfertile. And in the genome-wide association study, basically the investigators were able to narrow down the defect of infertility to a specific region of homozygosity in all those bulls. They all had the same homozygous region, and they all presented this mutation in a gene called TMM95. The mutation is in the transmembrane domain and supposedly prevents the localization of the proteins in the membrane. So Pablo in Madrid, Pablo Alvarez in Madrid, generated the knockout mice for TMM95, and he observed a similar phenotype to Izumo-1 knockout animals, meaning that knockout sperm accumulate in the perivitelline space because they are unable to bind and fuse with oocytes. This is true for in vivo and for in vitro fertilization, so males are completely infertile. And then we wondered whether TMM95 interacts with Juno and or Izumo-1. We tested the interaction again using our method that I mentioned earlier, and it's called AVEXIS. And we couldn't see any binding of TMM95 to Juno or to Izumo-1, while we have the positive interaction of Izumo and Juno in the assay to reassure ourselves that the proteins are functional and the assay worked nicely. So we couldn't detect any direct interaction, but then we asked, does the addition of TMM95 allows the cells to fuse to one another, so the sperm and the egg to fuse? And what happens if we put Juno, Izumo, TMM95 together on a population of cells? So we used an assay to detect cell fusion that we have developed here. Basically, we generated a population. We have two different populations of cells. These are mammalian cells. They express one half of the GFP, and the other population express the other half of the GFP, so that the cell population, when it's on its own, it doesn't fluorescence. You don't see any green signal. And also, one population express the full-length mouse Juno, and the other population express mouse Izumo. When you mix the two together, if nothing happens and the cells remain separated, you don't see anything, any green signal. But then when you had a fusogen like syncytin A, which is well known to induce cell fusion in mammals, you can definitely see green signals here. And also in the case of syncytin, there are very large syncytia being formed. So what happened when we added TMM95 to the assay? Basically, we couldn't see any green signal, indicated that even the addition of TMM95 together with Izumo and Juno is not sufficient to induce fusion. Again, this indicates that TMM95 is not involved, or is not the fusogen that we look for. And more recently, two other groups have published, they have published preprint in bioRxiv. They are not peer-reviewed paper yet. But basically what they showed is that they solved the crystal structure of TMM95 and also of another protein essential for fertilization, which is called SPACA6. It turned out that both proteins, TMM95 and SPACA6, have a similar fold to Izumo-1, specific in particular the N-terminal fold of TMM95 is similar to that of SPACA6 and to that of Izumo-1. And they also confirmed that TMM95 doesn't bind Juno. But in this paper here, they show that TMM95 actually binds to the membrane. They use human proteins, while we have used mouse proteins. And this raised the question whether TMM95 has a binding partner on the oocyte. And of course, which is the identity of this binding partner. Also, there is another question that we haven't answered yet, which is whether there is a co-regulation or an interaction, not direct, but still relevant, among all the sperm proteins that have been identified lately. There are data indicating that possibly there is this co-regulation. So there was a paper from Inoue showing that SPACA6, which is the other protein that is known to be essential for fertilization, is basically disappears in all the knockouts from Izumo1 and the other proteins that I didn't mention to you, but are again required for sperm-egg interaction. So it seems that not having one of these candidate also downregulates the expression of SPACA6. And the reverse was found in fish, where SPACA6 and this other protein that is called DCST2 are also required for fertilization. And it turned out that in the absence of SPACA6, also DCST2 is downregulated. So there are indications that there is some kind of interaction, even if there are the papers that have been published do not show any direct interaction between all these proteins. So coming back to the picture here, I hope I have convinced you now that we have a lot of novel candidates. The picture is much nicer than before, I have to say. So it's much more complete, but still there are a lot of questions that remain unanswered. And then I want to touch briefly on the polyspermy block, which I also mentioned at the beginning. So after fusion, of course, you know that the mammalian eggs, they establish a membrane block to polyspermy, which occurs before the modification of the zona pellucida. And it is established, actually, it depends on the species, actually. Some species are more efficient in establishing this polyspermy block. For instance, in rabbit, it's quite common to have polyspermy, but in mouse, it's definitely not. And what we observed is that June rapidly disappears after sperm-egg fusion, so immediately after fertilization. Here are two pictures of Juno-stained egg, showing that the signal for Juno disappears from the membrane immediately after fertilization and is definitely not longer present at the pronuclear stage, but even before that is not detectable anymore. We try to investigate and to see whether the protein was released on small vesicles, and this seems to be the case. So in eggs that have been ovulated but not fertilized, I hope you can appreciate the small dots here. This is a transmission electron microscopy picture. So the small black dots here are Izumo1. Are Juno on the egg membrane. And then after fertilization, we found the small dots localized in the perivitelline space in what appears to be small vesicles, but it would definitely need more detailed investigation to actually say that those are vesicles with membrane and not just single molecules shed from the membrane. And as for the mechanism that triggers this shedding, because we were really intrigued by this observation. As for the mechanism, we tried to dig out a little bit the mechanism, and we know that the polyspermy block at the membrane level is not established if eggs are fertilized by ICSI, so by injecting the sperm inside the oocyte, and also this block is not established when eggs are activated parthenogenetically, for example, by adding ethanol. So we checked what happened to Juno if we fertilize eggs by ICSI, and we found that Juno remains on the membrane of oocytes that are fertilized with ICSI. So indicating again that Juno shedding might be involved in the establishment of the O-lemma polyspermy block. So the model that we propose is this one, in which Juno and Izumo1 are essential for the interaction, the binding of the gametes. And immediately after fertilization, so after fusion, actually, Juno is released in the perivitelline space. And so achieving the double goal of acting as a decoy egg, binding the other sperm that might be present in the perivitelline space, and also, of course, by being released from the membrane, decreases the ability of sperm to bind to the membrane. Therefore, the shedding of Juno, we think it's really relevant for the establishment of the polyspermy block in mammals or at least in mouse. So again, I really wanted to conclude this part on the fertilization, leaving you with the many questions that we still have to answer. And I hope there will be more investigations coming up in the next years that might be able to answer these questions. And then the second part of the talk, which concerned another cell surface protein, always involved in reproduction, but not in fertilization. The protein is involved in the embryo transport in the fallopian tubes. So I have to say this project was quite challenging, and the real reason that we started to look at the knockout mice for this gene was that the mice were generated at the institute where we were before, and also the female were infertile, and the gene encodes for a cell surface receptor. So it looked like an interesting project, which was, but very challenging, I have to say. So the gene that we worked on is called ADGRD1, and encodes for transmembrane for a G-coupled receptor. This receptor, they have 7 transmembrane domains and a quite large extracellular domain, including autocleavage site. And also, as part of the initial screening of the knockout mice, there was this data here available already, indicating that the expression of the gene was restricted to the female reproductive tract, and in particular to the fallopian tubes of the mouse. So we started to investigate in more detail the phenotype of the female, and we went for the usual suspect. So we checked whether they were actually infertile. So we mated them with knockout and control animals, males, and females are completely infertile. We then checked whether they were able to produce eggs in normal numbers, which turned out to be the case. So they respond to superovulation normally. They ovulate a normal number of eggs. And then we checked whether those eggs could be fertilized. And the number of fertilization was exactly the same compared to controls, either in vivo or in vitro. So we could exclude at least this factor, this initial factor, so ovulation and fertilization. So we then wondered whether there was an inability of the uterus to sustain a pregnancy. So what we did was to check whether we could detect implantation sites in the female after mating, and in any case, we could never detect any implantation sites in all our knockout animals. But still, we wondered whether the defect was really due to something happening in the uterus. So to try and answer this question, what we did was we transferred embryos directly inside the uterus of the knockout female. And when we did that, we could find implantation sites, and the embryo could also develop normally inside the uterus of this knockout female, indicating that the uterus was not the site of the issue, basically. So we had excluded, based on the experiment that I've shown so far, we had excluded defects in ovulation, in fertilization, and in implantation. So there was just one site that remained to be explored, that was the oviduct of the mouse. Here is a picture of a stretched oviduct. Briefly, just a brief reminder of what happens here and how the transport works, because there is one big question that remained unanswered, and that was the unlocking of the ampullary isthmic junction. So basically what happens is that after ovulation, the eggs are catched by the infundibulum, and they reach very quickly the ampulla. But there they stay for a relatively long time, like 20 hours, until this junction is then unlocked and allows the passage of embryos from the ampulla to the lower segment of the isthmus, and then the embryo can continue its travel. And this long permanence of eggs in the ampulla is a typical event in all mammals. It happens in all species. So we thought we had to investigate a little bit better the mechanism of transport along the oviduct to try and understand what was going on here in our knockout animals. First of all, we checked after fertilization and after mating, a few days after mating, where the embryos were. So, and the surprising finding was that every time, regardless of the estrous cycles phase, I could always find oocyte and embryos inside the ampulla. Even at 2 or 3 days after mating, they were always localized in the ampulla. And also after mating, I think the nice thing is that they could really reach the morula and the blastocyst stage. And this was in agreement also with other finding from the 60s, in which they showed that embryos in the ampulla can develop and they can reach the blastocyst stage. So then we asked whether the defect that we were seeing, which was embryos are held in the ampulla, was an intrinsic defect of our embryos. So we transferred beads, glass beads of the size of oocytes inside the ampulla, and we checked what happened to them. But if you can see the video here, yeah, the beads move inside the ampulla where you have these contractions. They move nicely back and forth. And hopefully you can also appreciate the presence of 2-cell embryos here in the ampulla. This is a knockout animal. So the beads behave exactly like the embryo, indicating that it's not something that is due to the embryo, but the defect is definitely due to something going on in the oviduct. So we had excluded all the other possible explanations, and the only one that we could find for the infertility was that the ampullary isthmic junction was never unlocked in this female and never allowed the passage of embryos from the ampulla to the isthmus. We tried to, well, we investigated the mechanism in more details. So we took into consideration the different factors that regulate the embryo transport in mammals. There are 3 of them, the major ones. So the first one is, well, the ciliary beating. The epithelium of the oviduct is lined by ciliated cells. Their presence decrease when you move from the ampulla to the isthmus. So there are ciliated cells are less frequent in the isthmus compared to the ampulla. The opposite is true for secretory cells that also are present in the throughout the epithelium of the oviduct. The second factor regulating regulating the embryo transport are the muscle contractions. And the third one is the oviductal fluid. Muscle layers also change their distributions along the oviduct. So you have thickness muscle in the isthmus and thinner layers around the ampulla. So we wanted to test whether any of these factors were affected in our knockout models. So we started from checking whether the ciliated cells might be the reason why our ADGRD1 knockout mice were infertile. We checked the expression, the distribution of the cells, also their morphology here, and we could never find any difference. And also we checked whether the movement that they create when they beat were different. And again, we couldn't really detect any difference from our knockout compared to the wild type. So we excluded that ciliated cell were implicated in the phenotype that we were seeing. Then we checked the muscles. We checked again the morphology of the muscle along the oviduct, and we checked for the size of the muscle layers. And as before, we couldn't really see any difference. In here, you have in gray the wild type, and in orange the knockout. And there are no difference in the different regions, but what is nice is that you can see the difference in thickness as expected from the ampulla to the isthmus. So you have thicker layers of muscles in the isthmus of wild type and knockout. So we also excluded that the muscles contractions were that the muscles were involved in the phenotype. So the third possible factors was the fluid, and obviously measuring the fluid in mice is very, very difficult due to the size of the oviduct. We came across a paper published in 1968, actually, in which they were measuring the fluid of oviduct produced in sheep. And we were quite surprised by this, because basically what they show is that, so they cannulated, they put tubes and they measured from in the infundibulum and another cannula at the lower part of the isthmus. And they measured the amount of fluid that was coming out throughout the estrus cycles. And what they found was that there was a huge increase in the production of estrus, of fluid around estrus. And then there was a sharp decrease in this fluid. But what was surprising was that actually the fluid was not going towards the uterus, but it was moving towards the ovary in a adovarian direction, basically. So this is just a schematic of the direction of the fluid. So the fluid actually goes the other way compared to the embryo. So we wanted to check whether the production of the fluid, as I said, was somehow affected in the mouse in our knockout. And of course, this is quite challenging. At the time, there was another a paper from Yanagi-Machi being published, and we were taking more or less the same approach as them. So basically, as a proxy for the measurement of the fluid, we ligated the infundibulum here towards the ovary. We ligate the infundibulum and then checked what happened to the oviduct 3-4 hours after the ligation. What you can see is the expansion of the ampulla around ovulation. So these pictures were taken on the day of ovulation. And you can see that the ampulla is nicely expanded in knockout and in control oviducts. And then we checked what happened after that. And I hope you can appreciate that the ampulla in our control animals is not expanded as it was the day before. And it's also not expanded 1 day later, so 2 days after ovulation. This is not true for the knockout, in which the ampulla is expanded regardless of the time in which you ligate the oviduct, indicated that there was a dysregulation here of the fluid flow. So then to show and to try again to measure the fluid, again we take a similar approach to the one that Yanagi-Machi took in his paper. So we injected a dye in the lower part of the isthmus, and then we followed his path towards or along the oviduct. And from this still frame, I hope you can appreciate again that in the oviduct of a knockout, the dye immediately fills up the entire length of the oviduct, while in the wild type, the movement is different. So it basically, the dye is split up in small boluses, in small portions, and then it goes back and forth a little bit, and then it reaches the ampulla and the infundibulum. And these images were taken on a mouse 1.5 days after ovulation and after fertilization. I have a video here which shows the same in real time. So on the left you have the knockout, and as you can see, immediately after the injection, the ink reaches the ampulla here, while in the wild type it's still here going back and forth, and then at the end it reaches the ampulla, indicated that there is a dysregulation in this knockout that lacks ADGRD1 in the fluid flow. So for us, that was the demonstration that was the defect. And of course, because we are a lab that works on cell surface proteins, we wanted to know whether ADGRD1 had a binding partner. So we took a large screening assay approach again, and a collaborator in the US was able to identify PLX-DC2 as the binding partner of EDGRD1. We could show that the binding is conserved in human proteins and in mouse proteins between PLX-DC2 and EDGRD1, and we could narrow down the binding to two specific domains. The pentraxin domain of our receptor, EDGRD1, and a domain called PSI in the plexin protein. And quite surprisingly, we found that PLX-DC2 is expressed on cumulus cells. So based on all the evidence that I showed you so far, we proposed a model that entails that basically the presence of PLX-DC2 on the cumulus cells signals that embryos or oocytes have arrived in the ampulla, and then this allows the reduction of the fluid mediated by the activation of a signal that we don't know what it is, triggered by activation of EDGRD1 on the epithelial cells here. So cumulus cells signal to the epithelial cells decrease the amount of fluid that they produce, and then, of course, the fluid is reduced. This allows the passage of the embryos from the ampulla to the isthmic junction. And with that, I conclude my presentation and wanted to just acknowledge my PI, Gavin Wright. This was the place where we were before. They generated the mouse, all the knockout mice that we have been working with, and our collaborated with which I mentioned before, about the TMM95 project and the founders, of course. And thank you for your attention. Thanks.
Thank you, Enrica, for a very nice talk. Also, I will open the questions here.
Thank you. That was a real tour de force. Thank you. That was a wonderful talk. Okay, I think there's some really obvious questions coming up. Firstly, the use of a male contraceptive or just a general contraceptive blocking the Juno protein that's shedding is something that comes to mind straight away. Also, on the biological point, are these the only cells that actually are shedding Juno, or is it shed from somewhere else? Is it shed throughout the menstrual cycle and during times of infertility to help maintain infertility?
Yeah, so thanks for the questions. Juno is expressed specifically on eggs and only on T cells, regulatory T cells. So it's not expressed everywhere in the body. So far, and the role of Juno on T cells is actually not very well established. So far, I only know about the shedding of Juno from the oocyte at the time of fertilization. And as for the use of those proteins for contraception, I know there are, well, I think there are people working on it. We have done a little bit of work, but I think we still are a long way to go before we can develop anything to block the interaction for different reasons. First of all, these are surface proteins. They have large surfaces interacting with one another. So it's difficult to find something that blocks the interaction. And then you would need something that delivers that specifically at, well, in the female reproductive tract and has to reach specifically either the oocyte or the sperm after the acrosome reaction, because the ZUMO1 is not exposed before that. So I think there are many hurdles to be overcome.
Yeah, I have another question regarding your second part, the second part of your talk, and your knockout mouse. Did you actually look at the process of ovulation itself? Because the fallopian tube plays a very large role, important role in actually breaking the ovary and actually getting getting the oocyte out of the ovary and into the isthmus. Also, there's a lot of mucal fluid being produced there to make sure that the egg falls into the isthmus. Also, the amount of different components that are coming out in the antrum into the fallopian tube. Have you can you rule out that any of those processes are changed, and that's maybe adding to the infertility?
We haven't looked specifically at this, but because I could always find embryos inside the ampulla, I never suspected that there was something wrong going on there. I also looked at the timing of cumulus complexes disassembly. So basically, I looked whether the cumulus complex was maintained for a longer time in knockout compared to wild type, and I couldn't really see any difference there. Thank you.
Hello, thank you for your great talk. I loved both of them. Regarding the first, I'm aware that the fusogenic molecule hasn't been discovered yet. So I did my PhD thesis in gamete fusion, and that question was there like more than 15 years ago, and I feel it's still unanswered. Have you or the people in the field considered that maybe it is not a protein, maybe it's the lipid composition of the membranes or some other kind of molecule besides proteins involved in the fusion per se of gametes?
Yeah, thanks. Very, very interesting question. Great question. Well, yeah, the question is still there. There is no fusogen known in mammals. There has been a fusogen identified in lower eukaryotes, which is called UP2, works in plants and works in lower eukaryotes, but not in mammals. So yeah, whether there is or not a fusogen, whether that is a protein, I think nobody has the answer to. But before discarding the possibility, we have to explore it, I guess. And yeah, it's quite difficult to say no, it doesn't exist until, I don't know, we have screened the entire protein range.
Sure, but have anybody explored, for instance, the changes in lipid composition in the membranes of gametes at the moment of fusion to see if maybe that is changing or there's something about lipids?
Yes. So there was a paper published last year or 2 years ago showing that there is exposure of phosphatidylserine and that is required for sperm-egg interaction. So that's another possibility. Yes, sure. It's something they could work together because that work wasn't conclusive, I think. So we don't have a clear answer to that. So we don't know exactly what's going on.
Thank you very much.
Thanks.
Any other questions? If someone wants to do a question in Spanish, it's totally fine, the students. I don't know. No? I do have a question, but it's a technical question. How much follicular fluid are you able to recover? It's from the mouth. How many microliters? Must be very few. No?
I think Yanagi made a measurement. It says few microliters per hour.
Okay.
Which means that, yeah, it was really not possible for us to do a cannulation experiment like the one that they did in sheep, because you have to maintain the animal alive for a long time in order to be able to measure a very tiny amount. So it wasn't a way to go for us. It was quite complicated. I think it says like 2 or 4 microliters per hour.
Yeah, I was thinking something like this, very, very challenging to get it.
Yeah, it was.
All right. No more questions? No? Okay, well, there. Thank you very much, Enrica. It was a very, very nice talk, and I hope to see you soon. you know.
Yeah, possibly in person, yes.
Yeah, thank you very much, and goodbye. Have a good afternoon.
Thanks. Thank you, everybody. Bye-bye.
Bueno, con esto damos por terminada la sesión de la mañana, un poquito tarde. Los almuerzos van a estar en cajitas afuera y el lugar de almuerzo es...
Somos una universidad católica regional. Más del 80% de nuestros estudiantes provienen del Maule y más del 80% de nuestros egresados se quedan en la región. Somos parte del desarrollo del capital humano avanzado, fomentamos el capital cultural y generamos proyectos que van en directo beneficio de la sociedad. Desarrollamos el espíritu de servicio y el pensamiento crítico a través de nuestros planes formativos y sello valórico en pregrado, posgrado y educación continua. Somos parte de la historia de 15.000 personas que han pasado por nuestras aulas y hace más de 50 años que contribuimos con el desarrollo de la región, siempre responsable y comprometida con nuestro entorno. Contamos con programas de acceso inclusivo, porque creemos que no hay impedimentos ni sociales, económicos o físicos para concretar los sueños. La investigación que se genera dentro del UCM nace desde las necesidades locales. La innovación busca cambios positivos en la sociedad, dando respuesta a los requerimientos de nuestra zona. La UCM es fiel a sus orígenes, pero mantiene su vista en el futuro. Somos tu universidad.